thing to think about when we're talking about macromolecules is how is the structure of that macromolecule related to its function? And the same idea you can think about if you're thinking about what utensil would you use to eat a bowl of soup, right? So the shape, the structure of that tool or of that utensil would be the one that is fit for the function of eating soup, right? We were going to pick the spoon because of the shape. So the shape, the structure is related to the function. Okay, but also if we were to change that structure, that also means we're going to change the function. So if we were to take that spoon and put a hole in it, its function is now changed. So the structure is going to be related to the function. And if we change the structure, we change the function. Okay, so carbohydrates, we'll do a quick review. Remember, our monomer is monosaccharide. The polymer for carbohydrates is polysaccharide. The bond name is a glycosidic bond. The function is for short-term energy storage, and its structure is carbon, hydrogen, oxygen in a 1 to 2 to 1 ratio. Okay, so we're just going to go into some examples today. So we can have examples of monosaccharides. These are made of 3 to 7 carbons. You don't have to memorize that, but we should be able to look at one and identify that that is a monosaccharide. So notice we have carbons, and then for every carbon, we have 1 oxygen and we have 2 hydrogens is our 1 to 2 to 1 ratio. We're also going to have this carbon double bonded to an oxygen. That's called a carbonyl group. And their names are going to end in ose. So if we deal with anything, trioses, pentoses, hexoses, glucose, ketose, aldose, pentose, hexose, like all of these end in ose, we should be able to see a name and think, oh, that's probably a carbohydrate because of its name. Okay, we can have monosaccharides in different shapes. They can have linear, like these on the previous slide, or we can have ring-shaped molecules where they are bending around. So here it's the same structure. Here is there's another carbon that's assumed here, 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 and here. So that's the carbons in the middle, and then we have 1 oxygen for every carbon, and we have 2 hydrogens. Basically, that carbonyl is coming around to make our ring structure. Again, we just need to be able to look and see and tell that this is a carbohydrate, probably because there's so many oxygens and compared to our carbons. Here's some other examples. These are disaccharides. We don't need to memorize them, but just be able to visually look at the structure and tell that that's a carbohydrate. A disaccharide, di meaning 2, is made of 2 monosaccharides. So maltose, lactose, sucrose, all of these are examples of that. Polysaccharides, poly means many, so it's a long chain of monosaccharides. They can be branched or unbranched, and they can consist of more than 1 different type of monosaccharide. And a good example of this is something called glycogen, which doesn't have that wonderful ose at the end, but it is a carbohydrate, long chain. And this is usually how animal cells will store sugar. And so glycogen, you can actually see in this microscope image, these black granules here are the glycogen stores in the spermatozoa of a flatworm. So they can be, carbohydrates can be linear, they can have different shapes and rings, they can be branched, and glycogen is very branched. Okay, cellulose is an example also of a carbohydrate. It's got that ose ending. Cellulose has a different function, though. It is used in the cell wall of plants. So in this case, it's not a short-term energy storage. It's mostly used for structure of that cell wall. And it's going to have long cellulose fibers, which makes sense that they would be long fibers because that is what is giving that cell wall the strength. Okay, now we're going to move over to lipids and talk a little more about lipids. So remember the monomer for lipid is a glycerol and fatty acids. The polymer, we just call them lipids. The function of lipids is for long-term energy storage, but also structure and in membranes. Its structure is carbon, hydrogen, oxygen, and sometimes phosphorus. So here we have an image showing the glycerol and these fatty acid chains. Notice how many carbons there are and how few oxygens there are. Okay, so lipids are the big thing that defines them is that they are non-polar. Remember, that means they are not like water. They're going to have mostly carbon. They're not going to interact with water very well. Okay, so there are hydrophobic, water-fearing. So an example of this would be lipids that are found on the fur of aquatic animals, helping them be water resistant. So there's lots of different uses. Okay, there's saturated fatty acids and unsaturated fatty acids that can be used in lipids. The big difference between them, as you can see, is this double bond right here. So notice that when we have a double bond, we don't have as many hydrogens. So here for every carbon, we have 2 hydrogens, except on the end we have 3. Here for every carbon, we have 2 hydrogens. But on this double bond, this carbon only has 1. So we've lost the ability to bond to a hydrogen here because of that double bond. So the big difference between our saturated and unsaturated, our saturated have single bonds, is their state at room temperature. So saturated fatty acids, saturated ones will be solid at room temperature. I think of butter as an example. If you take a stick of butter and you leave it out of the fridge, it's going to still be soft. It might get, it's still going to be solid. It might get soft, but it's still solid at room temperature. It's called saturated because it's saturated with hydrogens. It's got as many hydrogens as we can possibly put on those carbons. Unsaturated fatty acids have a double bond, at least one somewhere, and it makes kinks because of that double bond. And what happens is that those kinks make it difficult for those unsaturated fatty acids to pack together, meaning it's harder for them to stay as solid. So they're going to be liquid at room temperature because they don't stick together as well. Okay, phospholipids, this is where I mentioned that lipids can sometimes have phosphorus. So a phospholipid is a lipid with a phosphate group attached to it. And notice that a phosphate group has a charge. It's got a negative charge, and that's going to be polar. And so that means we have a non-polar lipid attached to a polar phosphate group. So that's going to make it have a different type of structure and interaction with other molecules. Okay, so you can think about, here's a phospholipid. If it's got a hydrophilic, water-loving head and hydrophobic, water-fearing tail, think about how they would arrange themselves. The cell is mostly made of water, so there's water all around this molecule, and it's not going, the hydrophilic head would be fine, but the hydrophobic tail would not be fine. But there's more than one of these, so think about how this would arrange themselves in water. And there's different structures they can make. One can be these bilayers, so layers made of 2 layers of phospholipids, where this would continue in either direction. But we have water up here, water down here, but there's no water in the middle because of how they arrange those tails. We can also make these circular structures. So this would be like a vesicle, and we'll talk about this as well. These are basically these phospholipids are what's making the membranes for everything in the cell. The cell membrane, any sort of organelle we have in the cell, for the most part, a lot of them are going to be made of lipids themselves. Okay, now let's move on to proteins again. Remember, the monomer is amino acids, the polymer, polypeptides. The bond name is a peptide bond, and the function is for structure, movement, and energy sources, transport, immunity, all enzyme activity, all sorts of functions. And its structure is, we have an amino group with that nitrogen, a central carbon or an alpha carbon, and then a carboxyl group. And then we're going to have an R group off of that central alpha carbon. Okay, the big thing why proteins have so many different functions is because they have these different R groups. So that R is not an element because there's no R element on the periodic table, but that is basically a placeholder, and we can attach all sorts of different side chains, is what they're called, all sorts of different groups to that side. And these side chains can be hydrophobic, hydrophilic, or even ionic. So that means that they're going to then interact differently based on what side chain is present. So we can think about those hydrophobic and hydrophilic regions of our amino acids. How would they relate themselves to a lipid? So if we have a bilayer here, where would the hydrophobic amino acids be located? Well, they're hydrophobic. They don't like water, so they're going to orient themselves inside of that membrane. And then the hydrophilic amino acids, they like water. They're going to orient themselves outside of that membrane. They also could have these hydrophobic amino acids be located deep within the protein, and the protein fold to surround it. And we'll talk about that later. Okay, our last one that we're going to talk a little more about is our nucleic acids. Remember, the monomer are nucleotides, the polymer is RNA or DNA, the bond name is a phosphodiester bond, and its function is to store genetic information. The structure is a 5-carbon sugar, the phosphate group, and a nitrogen base. Okay, so we're going to talk about some differences between DNA and RNA, and this will come up many times throughout the year. So that sugar that we have is a ribose, and it's either going to be deoxyribose or ribose. So here we have a ribose sugar and a deoxyribose. So if you look at the difference in the structure, it's actually very small. The only difference, is this oxygen right here. There's an oxygen present there and not present here. So that tiny little difference is all that is changing between our DNA and RNA. But that one little change makes them vastly different molecules. Okay, but their main structure, that phosphate group, 5-carbon sugar, and nitrogen base, is still the same. We will just have one that's got a deoxyribose and one that has a ribose. And which one is which? Well, the deoxyribose will be the DNA and the ribose will be the RNA. Okay, so that's one of our differences between DNA and RNA. They also have different nitrogen bases. So the nitrogen base is located here. For a DNA, you can have adenine, guanine, cytosine, or thymine. So those are all examples of nitrogen bases. And then the RNA will have adenine, guanine, cytosine, or uracil. So instead of thymine, it's got uracil. But other than that, the adenine for an RNA is exactly the same as an adenine for a DNA. The strandedness is also different between DNA and RNA. DNA is always double-stranded. RNA is usually single-stranded. There are times where it can even loop back on itself and have regions that are double-stranded, but for the most part, it is single-stranded. And that's it for right now.